Hi, in this video I'm going to go through the poll questions for week 1 of 2024 fall. It's only a few questions. They're mostly about sequencing data and different types of sequencing. This first one is asking you to compare 3 different types of sequencing, specifically in the scenario where you're trying to sequence through a repetitive sequence containing region. The main thing to realize is that the problem with a repetitive sequence is that I don't know if it's completely repetitive, so this is a trinucleotide repeat. So if I'm somewhere within that repeat region, so 100 to 250 base pairs long according to my text here, I don't know where I'm at unless I touch sequence that's outside of the repeat region. And that's just because the inside of the repeat region is non-unique. It could be anywhere in there. And what's even worse is something like a trinucleotide repeat. There might be other very similar sequences elsewhere in the genome because they just kind of pop up from time to time. In fact, here I say that there are 5 of such repeats throughout this section of the genome. And so it's kind of a problem. And so let's go to the worst one here, and that is the 150 base pair single end read. And let's discuss why. I have a very professional, definitely not just MS Paint drawing to demonstrate this. So here is my genome. I can kind of scroll back and forth. I can see it's much larger. There's a couple of repeat regions indicated by red. Here's an example of a 150 base pair single end read. And in this scenario, I can see that the single end read has landed entirely within the repetitive region. So if I am trying to then map it, let's just pretend all I know is the data. Let's say that this is the actual position that sequence was generated from. I wouldn't know this, but let's just pretend that that's the truth, that it came from this particular nucleotide. If I take that ATG a lot of times, and I try to figure out where in the genome belongs, it could belong, let me get my little tool, it could belong here, it could belong here, here, it could belong over on this one, or this section, or upstream or downstream, anywhere that repetitive sequence is there, because it repeats. That's the point of calling it repetitive. It's the worst because if it lands within this region, so here I've made like a little diagram where I crossed a bunch out. Let me just remove that one altogether. If it lands within this region, I can't map it. And in fact, I would just preferably throw out that entire read. It's misleading. On the other hand, this paired end read, even though it's only got 75 base pairs on each end, now I did kind of make them add up to the same, so that way you wouldn't be trying to like balance things you don't really know. And honestly, it's hard to balance stuff like that. I don't have an equation just off the top of my head. I'd probably have to do some research to make that sort of decision, but I did make them comparable. I made them the exact same total amount of base pairs, 150 total sequence base pairs. The difference being this one has those base pairs separated by an ambiguous quantity of DNA. And so I say ambiguous is actually generated by sonic waves, which is pretty cool. It means that it's a little bit rough because it's not like we measured. It's just pretty close to a certain range of base pairs. But with this insert, we can actually map this sequence. If we assume that this region right here is unique because it's non-repetitive, and that's what it means to be non-repetitive, if it's unique at least, and unique enough, and that 75 base pairs is able to match, we can actually keep this read in the read mapping, and it can inform later analyses. It tells us things like this section of the genome is right next to a repetitive sequence. And if we have a whole bunch of these lined up, we can really start to learn a lot of information. So you start stacking all these together, and you start learning a lot about the genome, and we have algorithms specifically designed to take kind of the synthesis of all of these little paired-end reads and figure out how they best fit together. And while there may be still some ambiguity with that method of how long this repeat region is, you do start to overcome that issue with enough observations. Now that said, that still did cause problems, especially for larger repeats and especially for very kind of especially difficult to sequence repeats like an SSR, what we're visualizing here of ATG, also called a simple tandem repeat. The advent of long read technologies, especially high accuracy long read technologies, has really made such repeats no longer a problem. So in this one, this entire region is repetitive, and it's hard to sequence, and it's also difficult to map, but it's physically attached to other sides that are unique, and since we've sequenced the whole thing, we know its exact length, especially once we've really sat there and sequenced that fragment of DNA really, really well. That's actually the benefit of what's kind of the newest technology of circular consensus sequencing. That would actually be able to just sit there and just keep sequencing this fragment over and over and over until we were really, really confident that we knew the correct sequence for it, even though it does have that challenging to observe middle section. So that's the comparison. Because this one is so like perfect, it overcomes all the barriers, it's the best. Paired end reads is how we managed for a long time to get through regions like this. And then single end is the worst for this particular scenario. There are trade-offs with these, but for the most part, 75 base pair paired end reads would be better than the 150 base pair, as long as there is an actually unique sequence out on those edges. Now, if it's only somewhat unique outside of the repetitive regions, then maybe there's some ambiguity. But for specifically this application, that paired end read is probably superior. Now in real life, and at least in modern times, most of the time you're getting 150 paired end reads, with really the drawback, or the only drawback being the amount of time you have to spend on the sequencer and just the cost of getting that paired end read. Most of the time, you're just going to get the paired end if you can. But the long read, still kind of hard to get the sequence time on them, and still fairly pricey. And so that's kind of like the drawback here. Additionally, you do get more reads of this, but it does tend to balance out since these are, in fact, totally longer. All right, so the next generation versus Sanger sequencing methods. This is a massive difference. I saw lots of things. Some people were focusing in on the sequence length. It's not really so much about the length, although, in fact, Sanger sequencing tended to be longer than next generation sequencing. You'd get around 500 base pairs, where with next generation sequencing, even the best next generation sequencing, you're really not getting much more than 150 base pairs per side. So even then, the maximum is really 300 base pairs. Sanger sequencing definitely can go up to 500, technically even more, although I usually don't try to put much more than 500. The big difference between them is a Sanger method requires a single sequence many, many times inside of a tube of liquid. So one sample, one sequence. If you have multiple sequences, in fact, it's confusing because you get signals that are overlapping with each other. In fact, it's sometimes hard to do Sanger sequencing on heterozygous individuals because of that, because even a small difference can actually cause your sequence to be pretty difficult to understand. Not impossible, and there's a lot of those different strategies, but that's part of it, is Sanger method sequences one sequence at a time. Next generation sequencing, if you remember, we did that top-down view at one point where we saw all the little individual dots, tens of millions to hundreds of millions of different fragments, different sequences being imaged all at the same time, massively parallel. sequencing. Not just a little bit parallel, massively parallel. And so that's why I say it is an enormous, revolutionary difference. So yes, there are differences in like sequence length and efficiency and a lot of other things, but like the really big, crazy huge difference is that Sanger was one sequence at a time versus millions of sequences all at the same time. Okay, this next question is asking you to mostly parse this sentence and then apply it to the calculation that we discussed at the end of the period. Also, just a calculation, I want to reiterate, not a terribly challenging math. You're calculating coverage. Coverage is this concept of how much on average do you expect to see each locus within the genome, or at least which each locus within whatever you're sequencing within the genome. And so really what it comes down to is you simply need to calculate what is this is your numerator. I don't know why I'm doing this. Your numerator, we'll just circle it. Your numerator is the total amount of base pairs that is being sequenced. So how many total base pairs of sequences generated, divided by how much genomic space is that spread out across. And so if they're about the same, then you expect to see on average each locus once. If one is more, so if the numerator is more, let's say 30 times as much, then we would expect to see 30 each locus about 30 times. Once you get that concept down, deriving like how to go from stuff like this to the actual coverage is fairly trivial. Trivial, that's not a word. Trivial. It's about, all right, I have to just calculate how many total base pairs of sequence must be being generated. You read through this and you see that I'm going to generate 2 million paired end reads, and that each read consists of 50 base pairs per end. So 100 total base pairs per read. So that becomes 200 million total base pairs. That's it. The rest of that is just parsing my weird way of structuring this, sorry, and figuring out how to enter in the answer. But in the end, it's just calculate this numerator needs to be 200 million in some way. And by the way, if you did actually get the correct answer, I think I managed to limit your choices. I did manage to limit your choices, so that wasn't possible. There's only one solution to get 200 million on the numerator. And then Z is just going to be the size of the genomic space. In this case, that's 0.7 gigabase. 0.7 giga is going to be 3 orders of magnitude above mega. And mega is million, which most of you can figure out is 6. This is how I have to do this same thing. I don't know why. I also have a block on giga. Giga is to the 9th, but I can't memorize that. But I can memorize that mega is million is 6, and then I go 3 above it. That's just how I work it too. But in the end, that's what you're going to pick. So you're going to have to pick. So either this and this, or this and this. I can't remember if I actually. Yeah, I didn't stop. So either way would work. But it's got to be 100 base pairs times 2 million reads. And then it's got to be 0.7 giga. That's it. The total amount of base pairs divided by the total base pair space it's spread out across. Not too bad once you kind of get your head around it. Okay, that's it. I hope that helped. Please reach out if you have any questions. I don't generally read the comments, so because I'm a smart internet user and I don't read the comments. Anyways, y'all have a good day. Bye-bye.